Okay, folks, we're on the last lecture of Unit 1, which is woohoo, yay, excellent. However, it's probably also going to be our longest PowerPoint of the unit. So make sure you're nice and comfy and make sure you've got a good block of time and try to, even if you don't listen to me twice, try to maybe go through the PowerPoint twice. There's a lot of information here and a lot of new terms. So far we've been looking at, or we've been referring to membranes sort of, in a very casual way. We were looking, or you were looking at a review of a lot of your organelles, cellular organelles, many of which are made of membranous coverings, sort of, the whole structure is based on membranes. But we haven't really looked at what membrane is. Maybe in your mind, you might think of it as something that's sort of inert, something that's just like a plastic bag kind of thing, in the sense that it doesn't really do very much, or that it isn't made out of anything that's varied or diverse. And that's absolutely not true. So we're going to take a closer look at membrane structure, and then we're going to be looking later on in this PowerPoint at how things are able to move across the membrane, what the criteria for that is, and ways that the membrane itself is able to bring in larger things, or to allow larger things to leave the cell in quite complex acts. So first, for membrane structure, the sort of main components of a membrane, not the only ones, but the main components are a lipid and protein in nature. And of course, we've mentioned when we were doing our biomolecules that phospholipids are going to be an important component of the cell membrane. And one of the reasons for that is because phospholipids have the hydrophobic tail and the hydrophilic head. We call those kinds of molecules with that dual nature amphipathic. So part of the molecule loves water and part of the molecule hates water. So if you have a look at the graphic on the side, you can see the top of the molecule that's here is composed of a phosphate group and the part of a glycerol molecule. So that gives us, or gives the phospholipid its polar head. There's a lot of oxygen and hydrogen up in that top part. And then down in the bottom, which is sort of in the white paneled part of the graphic, we can see there are two fatty acid chains. The two chains that are illustrated here are saturated chains, so there's no kinking, but we'll see later on that there's a variety of fatty acid chains that can be here, and depending on what chains there are, we're going to see a little bit of difference in the way that they work within that membrane. So phospholipids are the primary, the sort of basic structure that we're going to see in a membrane. Now, because of the nature of the phospholipid, the fact that there's a polar end and a nonpolar end, we're going to see that the phospholipid has to work as a bilayer, particularly because both inside the cell and outside the cell are primarily made of water. So as you can imagine, the hydrophilic head is fine with that, but the hydrophobic tail not so much. So the way these line up is in such a way that the hydrophobic tails are together away from the water, and the hydrophilic heads are apart, both facing either side of the sort of watery fluids. So the hydrophilic heads are exposed while the hydrophobic tails are tucked away. We can have a look at this graphic, and we can see in this particular example that the hydrophilic heads are blue and the hydrophobic tails are sort of gray-white. And again, hopefully you can see that this is a bilayer, so a double layer where the heads are facing out and the tails are facing in together. Not only the phospholipids are amphipathic in a membrane, but also the proteins. And again, maybe think of this a little bit while you're kind of mulling over the diagram, if the protein is sitting in the middle part of the bilayer, so if you have a look at what's labeled on this diagram as integral proteins, the purple swirls, you can see the coiled part of them is running through the hydrophobic region of the bilayer, and that the more loosely curled pieces are running through the hydrophilic heads and then out into the fluids on either side of that cell membrane. So the protein here is amphipathic as well. The portion that's in the center has hydrophobic side chains that are fine in that lipid environment, and the side chains that are found on the parts of the protein that are outside of that, so sort of through the hydrophilic heads and out into the fluids, are in fact in some way hydrophilic. So they could be, again, could be hydrogen bonding, could be ionic, covalent, etc. So if you were to take this one of these squiggly purple proteins and try to poke it down through from one side of the membrane to the other, and then you moved your finger back, if that was possible, or some kind of a probe, poke it, the protein would pop right back into the same position because of the nature of that amphipathic nature of the protein. So it wants to be, or it is going to be situated like this. The phospholipid bilayer is fluid. It's not a solid structure. So the protein could push through if it wasn't for this, this sort of hydrophobic, hydrophilic nature. It could push straight through. So membrane proteins rather are amphipathic. And the examples, the longer purple examples here, have two ends that are hydrophilic and then the center that's hydrophobic. But if you look at the smaller purple ones, sort of in the middle there, they've got only one part that's hydrophobic and one part that's hydrophilic. And you can see the integral proteins are going all the way through where those other proteins are not. So proteins are regularly embedded in the phospholipid bilayer. The way that they're embedded, or the order in which they're embedded, or how many there are, what types there are individual to the kind of membrane that it is. So it gives character to the membrane. They all have the phospholipid component, but the membrane gives the functionality of that membrane and the individuality of the membrane. So, as I'm mentioning, not all membranes are identical. They are going to be quite different in their functioning. And the model that we use to talk about membranes is fluid mosaic model. And that's what I was speaking about earlier, the fact that the proteins and other components of the membrane are basically sort of the word I used in the PowerPoint here is bobbing in the fluid bilayer of the phospholipids. So it's almost like the phospholipids are forming, if you can imagine pouring oil on the surface of water, and we get that layer of oil. And if you could take, so that would be like your phospholipids. And if you could take a pencil, you could push that pencil all the way through that layer into the water, and you can move the pencil around in the water. And the oil will sort of fill in behind it. And that's sort of like the proteins in the phospholipid bilayer. And here's another representation of that. In this case, the hydrophilic heads are yellow and the hydrophobic tails are green. And we can see that there are some proteins that are going all the way through, in this case labeled transmembrane protein. And the part of that protein that's sticking up with the hydrophilic heads on either side of the bilayer, those parts of the protein are going to have to also be hydrophilic. And the part of the protein that's in amongst the green hydrophobic tails is also going to have to be hydrophobic, and it's labeled hydrophobic region here. And then you get some proteins that would be sitting just on the outside, and they, again, might have just one hydrophobic part and one hydrophilic part. The membrane itself is held together by those hydrophobic interactions. So the fact that there is water on either side of the membrane is in part what holds it together. If it wasn't for that, if there was fat soluble material there, like oil, if we had extracellular fluid that was oil, then we might have some trouble keeping our membrane structure the way that that we need it to be. And again, because it is because the membrane is lipid in nature, the membrane is fluid, just like that idea of the oil on the surface of the water that I mentioned a moment ago. So, as you may or may not know, some oils are going to, pardon me, be more or less fluid. So think about the difference between, say, a vegetable oil and something like coconut oil. It's very different. And how fluid the membrane is, a structure is related to a couple of different things. One of those is the kind of fatty acid chain that the phospholipid has. As I mentioned a little bit earlier, if you have unsaturated fatty acid chains, you're going to have a little more fluidity. And the other major component that impacts on membrane fluidity is cholesterol. And the more cholesterol you have, the more fluid your membrane is. And again, in this little diagram, we're showing some of the movements or some of the things that might happen with the phospholipids. So the first scenario A here, we're looking at movement of phospholipids, and we're learning here that lateral movement is quite frequent, and that's what I was trying to sort of illustrate with the pencil in the oil layer, and sometimes the oil layer rather, and sometimes you do get this flipping, but that is a very rare occurrence because of the water on either side of the membrane. If you look at the B scenario here, we're looking at one membrane on the left that is very fluid because of all the unsaturated fatty acid chain, all of the unsaturated fatty acid chains in those phospholipids, versus a membrane on the right here that would not be as fluid because the fatty acid chains are mostly saturated and therefore quite a bit straighter and not as fluid. And at the bottom, we can see to add fluidity to a membrane, we might have added cholesterol. And we can see our four-ring structure here representing cholesterol, and that the cholesterol will add some fluidity. And I think I mentioned in class that if you looked at polar bear membranes, they're going to be much higher in cholesterol than human membranes, and that is one of the adaptations of the polar bear that would make it better suited for the northern climates than we are. All right, so as I mentioned before, the combination of proteins is what's going to make the membrane unique, because the phospholipids, other than the saturated or unsaturated fatty acid chains, are pretty much the same from membrane to membrane. The proteins that are in there can be anchored or they can be fluid, so they can actually move laterally. Again, they're not going to move so much up and down, if you like, because of the amphipathic nature of the protein. If you look here, we can see that some of our proteins are sort of anchored in the microtubule cytoskeleton of the cell. So in this particular diagram, we're seeing it would be the inside of the cell would be to the bottom of this slide, and the graphic, and the extracellular fluid is at the top. And some of these proteins are integral proteins, meaning they go right through the membrane structure, and some of them would be what we call peripheral, and we'll see definitions of those in a little bit. Right here, in fact. So proteins that penetrate the bilayer are called integral, and ones that are only sort of on one surface or the other are called peripheral. So they're hanging on sort of to the outside of the membrane. And each of these proteins are going to have a side chain structure so that they fit or they sit in the right position in the membrane. And that's going to mean that they have hydrophobic regions and they have hydrophilic regions that require them to be submerged into the fatty acid chains, the sort of hydrophobic region of the membrane, or not to a certain degree. All right, so if we've got all those proteins in these membranes, there must be a reason for them to be there. And here is a pretty good overview of some of the ways that protein can be useful in a cell membrane. So they can be used for transport, and we're going to see a lot of that in a few minutes. So we can use some of those integral proteins as protein channels for moving things from one side of the cell membrane to the other, and we do that quite a lot in our cells. They could be an active site, an enzyme that might be there as an embedded multi-enzyme complex, something like that. So that substrate will come in to the active site and will leave, and so on. And we did talk a little bit about multi-enzyme complexes already. It can also be some kind of a signal. It could be like the one showing here, where a message goes through which causes some sort of change to occur inside or outside of the cell, or it could just be able to receive a particular message on the outside, which causes some kind of change on the inside, like say insulin, which is going to change the cell's permeability, the cell membrane permeability to glucose. So that's, sorry, I'm keeping up with that's the signal transduction, so it can be used as a receptor for info from the outside or the inside. It can be a location for cells to link together, so kind of hook things together. And it can give some recognition. Basically, we haven't looked yet, but we will look at carbohydrate that we find sometimes in the cell membrane. And we each have a unique pattern of carbs that are usually hooked to proteins. So we have what we call a glycoprotein. We usually have a unique signature. And our cells can recognize that they are our cells by that signature. Same with our immune system. We can recognize our cells as our cells by that glycoprotein signature that we have on the outside of our cell membranes. And they can add some structure to the cells. Again, we already mentioned the idea that we can have proteins attached to the microtubules that form the cytoskeleton of the cells. So carbohydrates are found often on the outside face of the membranes, and this little sidebar here, this little graphic is showing us how that might be the case. In this graphic, we're seeing carbs, the little green structures, being produced in the protein and glucose structures or sugar structures being produced partly in the endoplasmic reticulum and then packaged and sent out from the Golgi apparatus. And we're seeing actually exocytosis, so it's being removed from the cell, and then the carb portion is being distributed on the outside, or we're seeing that they're sort of being incorporated into a cell membrane as a signal, a signature to show that the cell is our cell. And the carbs can be attached to lipids or proteins, and we see, or we would see if we looked, that often there are more glycoproteins than glycolipids, and they do enable recognition. So enough about structure. We're going to move on to how the membrane controls what goes in and out. So two primary kinds of, the two sort of big categories for this kind of transport across the cell membrane, whether it's into or out of the cell, are called passive transport and active transport. So passive transport requires no ATP, while active transport does. That's one of the big differences. Because no energy is required in the passive transport, that means that we are moving with the concentration or down the concentration gradient. So we're going to be moving materials from an area of high concentration to an area of low concentration. That's not necessarily the case with active transport. We're usually moving things against the concentration gradient. That's why it takes energy to move those things. There are three kinds of passive transport and two kinds of active, and we're going to look at all three of the passives and two of the actives. Diffusion, osmosis, and facilitated diffusion are passive, and we have active transport primary type and we have active transport secondary type that require ATP, and we're going to look at those as well. So diffusion is what we call the process where molecules, ions, etc., that are dissolved in the extracellular fluid or in the cytoplasm of the cell are going to be moving into the cell, out of the cell, and so on. This motion is related to the concentration of these materials and is sort of an ongoing thing. Generally what will happen is that the molecule, the ion, whatever it is, will move from an area of high concentration to an area of low concentration until both sides are equal, so equilibrium is reached. When we're talking about diffusion, we're generally not talking about water. We're usually talking about some other substance that's being moved in the cell. Diffusion rates across the membrane can be affected by several different things. One is molecule size. The bigger it is, the harder it is to push through the membrane itself. Also, quite a lot of things that would be moving across the membrane may have some kind of polarity. And you can imagine as soon as it hits that phospholipid bilayer, which is hydrophobic, that's going to be a little bit difficult. It's going to be difficult for them to get through unless they're traveling maybe through a protein, which is what we call facilitated diffusion. So it's not going to necessarily move passively if it is polar. And a molecule or ion is charged. So not just polar, but also things with a charge for the same reason are going to have difficulty getting through. And here's a little graphic that maybe helps to illustrate that. So oxygen, which has basically no polarity and no charge, is able to move through the cell membrane very easily. Carbon dioxide, which has some polarity to it for sure, but is really teeny, is also able to go across the membrane. And water, which again we know is polar but is teeny, so can often slip through or can slip through the membrane. So we can have movement of those molecules across the membrane. Again, carbon dioxide and oxygen are good examples of diffusion. When we're talking about movement of water, we're usually talking about osmosis. So water can move freely through. Again, even though it's polar, it's teeny tiny. So all three of these things come into play. If it's polar but not too big, it can still probably go across. If it's really large but only a little bit polar, maybe it can't go through. So it's a combination of those factors. When we're talking about osmosis, we're generally talking about movement of water across a membrane. So we're not thinking so much about moving the material that's dissolved, the sugar, the salt, or whatever it might be. We're talking about the movement of water. Often the movement of other materials will follow, but when you think of osmosis, think about water molecules. Now here we're seeing a graphic where they're showing a semi-permeable membrane, so a membrane that will let some things through and not other things, and that's generally what we find with a membrane. And on one side of so on the left-hand side, we have high solute concentration. That means we've got a lot of stuff dissolved in the water. And on the other side, we have a low solute concentration. So don't let have a look at the blue dots in there, which is the material, whatever it is, the stuff that's dissolved in there. Let's say it's sugar. But look at the blue, or at the red dots. So the red dots are representing water. Look at the concentration of water on the low solute side. It's much higher. So the solute concentration is lower. That's the dissolved particle is lower, but the concentration of water is much higher. If you look at the high solute side, there's a lot less water. So the concentration of water is lower. What's going to happen here is water will move from the right-hand side to the left-hand side. Again, we're moving with the concentration gradient, not the sugar one, but we're moving with the water concentration gradient. So we're moving from high water concentration on the right to low water concentration on the left. People get that confused all the time. Make sure in your mind you've separated out the difference between the solute and the solvent, which in this case is our water. All right, so again, it's the concentration of, you have to keep in mind the concentration of all the components that are in whatever our solutions are. And often when we're referring to that, we may be referring to what we call the osmotic concentration. So the solution, whichever that might be, with the higher solute concentration is hypertonic, meaning that it has more stuff in it, more solute, so more sugar or whatever. And the solution with a lower solute concentration is called hypotonic. People have so much trouble with this when it comes to tests. Just keep in mind that hypertonic and hypotonic refer to the stuff like sugar and salt that's dissolved in the water, not to the water itself. Very important. If they have the same, if both sides of a membrane, for instance, have the same concentration, then they will be what we call isotonic, which means equal. So if we have a look here, our solute molecule is pink and our water molecule is clear or white. And the top in the top part of the graphic, we can see an isotonic situation where there's a membrane in the middle and both sides have equal solute concentration. And if we look at the bottom one, we've got a membrane in the middle and we've got a hypertonic solution on the right because there's more solute dissolved in there. And we've got a hypotonic solution on the left. If we were looking at movement of water here, we would see that water is going to be moving from hypo to hypertonic, from high water, low solute concentration, to low water, high solute concentration. That is the way that water will move. Here's an example using red blood cells. So when we look at animal cells and they're put in certain solutions, water is either going to enter the cell or leave the cell by because of this osmotic pressure. It's going to move due to osmosis, again, unless we have something that is isotonic, in which case you're going to have water moving in and out in an equilibrium situation, equal water in as water out. So if animal cells in particular are placed in what we call a hypotonic solution, meaning there is a low solute concentration, high water concentration, versus the cytoplasmic fluid in the cell. And again, we can only have hypotonic, hypertonic if we're comparing to each other. Something that may be hypertonic, or a concentration that may be hypertonic in one situation may be hypotonic in another. These are not absolute designations. It's only in reference to each other. So in this situation on the left here, this regular red blood cell was placed, let's say, straight in pure water, or maybe distilled water, or in distilled water with a very little bit of salt dissolved in it, compared to the fluid inside of the red blood cell, which would have a higher solute concentration, salts and so on, and a lower water concentration, compared to the outside where there's a high water concentration and a low solute concentration. Water will stream into that cell. And because of the nature of animal cells, where we have no cell wall, quite possibly the cell will burst, and we call that lyse. We will see that the cell has lysed, or we could say that lysis is occurring, or that it has been lysed. And in the middle situation, we've got an isotonic environment, and again, water's moving in and out, and so we don't have to worry about the red blood cell exploding or shriveling, like we're going to see in the final panel here, where this red blood cell has been put into a solution where there's a lot of dissolved concentrate. So there's all kinds of salt and sugar and all kinds of other chemicals in the liquid, and the cell's been put into that, and so there's a lot higher water concentration inside the red blood cell than there is outside. Water will move out of the red blood cell and it will shrivel. In an animal cell, crenation is a word that's used to describe that shriveling. Things are a little bit different in a plant cell because of its cell membrane. And if we do the very same thing that we did in the previous panel, where we're putting cells in particular kinds of solution, we'll see already that there's quite a difference in what's happening. Not in the movement of the water, direction of movement of water, but just the impact that it has on the plant cell versus animal cell. So if plant cells are placed in a hypotonic solution, meaning low solute concentration and high solvent or high water concentration, water will flow into the plant cell, and a lot of that will be stored in the central vacuole of the plant cell, and the inside of the cell will swell and will push against the cell wall, that rigid structure of the cell wall. And because the inside part is pushing against the outside part, we get a nice rigid cell structure, which we would call turgid or turgid, and would give us what plants prefer. They like to be able to use this sort of water skeleton effect to stand up nice and tall. So they don't have to worry about bursting because the cell wall will push against or apply pressure against that, and the two together are able to keep the plant upright and healthy. If a plant cell is placed in an isotonic solution where we have equal water in as out, we may have a plant cell that's a little droopy. So not enough water, time to water that plant. It's okay, but it's not ideal. And then if we place the plant in a liquid that has a lot of solute dissolved in it, so maybe a salt solution, we'll find that water will move out of the plant cell and into the environment. And in that case, we are going to see that the inside part of the cell sort of shrinks in on itself. And rather than being called crenation, we have in plant cells a term plasmolysis when that happens. So absolutely not ideal for your plant, probably on death's door. So still working with the concentration gradient, so still looking at passive transport, no ATP required. Sometimes, as I mentioned, for those polar molecules or charged molecules, because of the charge or the polarity, along with the size and so on, it may not be able to make it through the cell membrane, through the phospholipid bilayer. So instead, they can be carried through the membrane via a protein. So it could be something like we see on the left here, where we've got a nice little protein channel and the material is moving from high concentration to low concentration. Or there could be something a little bit more elaborate, a little bit even more specific perhaps, where we have a carrier protein that is able to change shape, kind of like an enzyme, when the substance enters it and allows it to be moved to the other side. Both of these will require no ATP at all and are working with the concentration gradient. So moving material from a high concentration to a low concentration. And again, it would only be used, or it would be used for things that are too large or too polar to go through the bilayer. And here's a little a panel telling us a little bit more about the channel protein and the carrier proteins. And again, something that you may take note of here is that the inside of these proteins are going to have to be hydrophilic to allow the polar molecule or charged molecule to go through, where the outside part of the protein is going to have to be amphipathic. We're going to have two ends that are hydrophilic and the center part that is hydrophobic, so that it can sit in the membrane at the right level, so that it's not going to move up or down in the membrane, even though it can still move laterally in that membrane. Carrier proteins are a little more specific and work a little bit more like an enzyme, as I mentioned, and they can be used to transport larger polar molecules like glucose and amino acids in and out of the cell. And again, in this case, we're moving with the concentration gradient from a high concentration to a low concentration. All right, now we're going to move into the kinds of transport that require ATP. What we're looking at here is things usually moving from a low concentration to a high concentration. So for some functional reason, the body is trying to concentrate material in a particular location, so it must be moved that way. And obviously our energy source for this is going to be ATP. It's a nice little graphic of ATP on the side here. I like it because it shows ATP sort of as a rechargeable battery. If you look at ATP at the top, you can see that when it's being used, so the gray arrow showing energy available for cellular work and for chemical synthesis, you can see that one of those phosphate groups is being hydrolyzed off the ATP molecule and used to do some work transferring that energy. So we end up with ATP with 3 phosphates, or we start with, ATP with 3 phosphates, and we end up with ADP, which has 2 phosphates. That are basically this molecule is less energy rich, and then we can use the process of cellular respiration or photosynthesis even to make some new ATP by grabbing enough energy to force that very negative phosphate group on the end and reform ATP. So rechargeable battery, ATP, use the energy, ADP, add some energy, ATP. So a cyclic arrangement. And again, in this case, we're moving against a concentration gradient, so requires ATP. So primary active transport and secondary active transport are really very similar. In primary active transport, the cellular process that's moving the molecule or whatever it is, ion, from one side to the other, is able to directly use the ATP. And the diagram here, we're showing a sodium-potassium pump. There is a location for phosphorylation, which will change the shape of this molecule and force the material in the direction that is against the concentration gradient, so from a low concentration to a high concentration. Cool thing about sodium potassium pump, a dual channeled apparatus or setup or protein here, is that it moves sodium in one direction at the same time that it is able to move potassium in the other direction. Secondary active transport is very much the same, but sometimes we have sort of a step that has to happen first that is able to then help the whatever the primary action is occur. So that sounds more complicated than it really is. This diagram is pretty good at showing that. If you look, we can see that ATP is being used to concentrate hydrogen ions, so moving from low concentration to high concentration. That's where the ATP is being used. So that would be primary active transport. And then that hydrogen that's being pumped across, so it's a hydrogen pump, being pumped across is being used to couple with sucrose and move against the concentration gradient for sucrose. So for hydrogen going through the red channel protein here, that is actually passive, right? It's facilitated diffusion going from high concentration to low concentration, but for sucrose, we're going from low concentration to high concentration. So the hydrogen is helping the sucrose move along. There's no direct ATP use in that red channel protein where there is in the yellow one. So it's kind of like one step helping another step occur. That would be secondary active transport. The very last thing that we're going to look at is membrane-assisted transport. And as the title might give us reason to believe, we're looking at what can happen with things that are maybe being manufactured or that are available in large amounts. And we actually use a piece of the membrane to move things across. Now, we actually already saw that in our little example at the very beginning where we were looking at moving glucose or sugar, signature sugar molecules out of the cell so that we could use them to identify our cell membranes as our own. So we're going to be looking at things that are sort of being bound in membranes and then being transported either into the cell or out of the cell. So the cell itself is going to form vesicles. Either we're going to form them from the outer cell membrane or we're going to form them in the Golgi apparatus, and we're going to use that to move things around. This also requires energy. So it is an example, membrane-assisted transport is an example of active. So endocytosis is a neat little thing that our cell membranes can do. We sometimes see this in video of single-celled critters like amoeba, where they're able to surround a food particle that's out in the environment with their own cell membrane, kind of extend pieces so they look like little arms. Pardon me. So by engulfing the particle with these little wavily arm things, the food particle is drawn into the amoeba and then is coupled usually with a vesicle of some description that contains some kind of digestive enzyme, and then digestion of that material will occur. So it has been called endocytosis, has also been known as cellular eating. Our cells are able to do the same thing, not quite as dramatically as an amoeba, but we are able to pinch off pieces of our cell to bring in solid material or liquid material, and they're given a distinct name here: phagocytosis for bringing in solid particles, and pinocytosis for bringing in liquid particles or things dissolved in liquid. The last panel here from our graphic is a little bit more complex. It's receptor mediated, so there's actually got to be the correct receptor on the cell membrane. They're showing it illustrated here as a coated pit, and the material must actually bind, and then a vesicle will be formed, and the material will be brought into the cell. So it can be a little more general, or it can be a little more specific depending on exactly what the material is and what the needs of the cell are. So there they are: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Exocytosis is the reverse, where we have material that's already been packaged. And again, in this graphic, we can see that we've got a secretory vesicle or vesicle that's been made from the Golgi apparatus. It's going to migrate toward the cell membrane, and because of the fluid nature of the membrane, it's able to sort of become part of the original membrane that's already there, and the material that was packaged is released into the extracellular fluid, whatever that might be. This could be maybe something that the cell has manufactured, like an enzyme or a hormone, and we could be dumping it into a digestive tract or into the bloodstream, or it could be a whole lot of different things that the cell may be preparing for export for neighboring cells or for other parts of a multicellular organism.